Okay, so this video is to briefly cover the signaling transduction pathways that we covered in the beginning of the class, but hopefully as you're kind of paying attention throughout genetics and what we did today, which was actually seeing how transcription factors, repressors, activators, how they affect what genes turn on and off, those are controlled by pathways like this that receive incoming signals. So first things first is that you have a receptor here, and it's got an outside area, so an outside portion of the domain, and an inside portion of the domain. So this is one whole protein. It's in a big 3D shape. Now it's obviously not going to look nice like this Y. It's like a big globe of amino acids basically. When you get your signaling molecule, which is your ligand, this could be a neurotransmitter, could be a hormone, could be an ion sometimes, basically anything. Hormone is probably the best because it's a good example of how a tiny molecule like this can elicit this massive response. So this ligand is floating around through the bloodstream. Let's say that it goes in and it binds here. Now it's only going to bind to the receptor that it can actually hook onto at specific amino acid points. What happens at this stage is that when this gets bound, there's a bunch of changes throughout the protein because you've bound up certain connections between amino acids. Now the 3D shape is going to kind of change. So instead of this inside shape that is off, the changes essentially elicit the formation basically of an on-site. So imagine that now this is kind of opened up. And now that this is on, after the ligand was bound. What's on the inside are these cascade proteins, usually kinases. That's what I'm going to call kinase number 1. What kinase number 1 does is it's just kind of hanging out here. It's not really doing much. But the minute that this turns on up here, it heads in. So kinase 1 binds to the on version of the receptor, which has been triggered by the ligand. Once it's in this conformation, it's actually able to accept a phosphate, and this usually comes from ATP, and there's some kind of enzyme help that's going to add this phosphate to kinase 1. Now, the only way that kinase 1 gets activated is if this was in the on stage. So now it's been activated by adding a phosphate. So if we go down here, we have an active K1 because now it has this phosphate. The phosphate changes the shape of K1 to an on state. So over here, it's off. It doesn't have the phosphorylation. Phosphates are huge, and what they do is they essentially disrupt the 3D shape of a kinase. They change it to a different shape. That's the on shape. The role of kinase 1 is usually to find kinase 2, and what it's going to do is it's going to find another ATP molecule, so a new phosphate. It's going to add a phosphate to K2. The role and the purpose of K2, now that it's been activated and turned on by the phosphate addition, because now you have changed conformation and a changed structural change of this protein, so it looks different, it's now on, is to find K3. What it's going to do is basically add a phosphate here too. It's going to turn it on. This is a new phosphate from a new ATP. So ATP is being added at each of these. So remember kinase phosphate or kinase enzymes, they're basically adding phosphates. Now you keep doing this. So remember, you've unlocked kinase 1. Kinase 1's role is to unlock kinase 2. Kinase 2's role is to unlock kinase 3. So on and so forth. You can do this multiple times. This is where we amplify the pathway. Because remember, at your ligand and receptor stage, you have 1. You may have 10 of these K1s that bind up. You may have 100 of these K2s that are affected by K1s. And who knows, after that you may affect 100 molecules at a time, and you keep adding and adding molecules downstream as you go. And that makes the pathway basically Basically, it cannot be shut down if you had this many molecules amplifying the signal. So the pinnacle, and let's say that K3 is the final pinnacle. What an active kinase 3 can do, we'll just do this little bubble as the phosphate. What it can do in a lot of cases is activate one of the molecules that we saw today, the transcription factor. What this can do is, in some cases, all it has to do is maybe add a phosphate. Now this is on. We saw today that transcription factors are key players when it comes to activating genes. So in this case, let's say that this DNA is off because there's no transcription factor to bind it. It's not active. Once you've added this one here, now this is active. Now it is binding the DNA, and the DNA is going to switch from off to on with the help of the transcription factor. And what this is doing is initiating transcription, and it brings in that RNA polymerase. So this is the typical set of steps that we outlined. Reception, transduction, and eventually transduction affects something, possibly a transcription factor, that causes a genetic change that leads to a physiological change. Not all genes are active all the time. They usually depend on some kind of system, like a ligand coming from another cell to communicate, things like that turns on a transcription factor, turns on a gene, the gene starts having the effect. So, if I asked you how this could be different in different cells, you should be able to create all kinds of different ways that this pathway could affect something differently in a different cell. Because remember, you have neurons, heart cells, muscle cells, skin cells, intestinal cells, all kinds of stuff. The same ligand is not always going to trigger the same effect, even if the pathway is similar. Let's say, for example, what you could actually do, say in some cases this is going to activate a gene. Let's say that in a different cell, this K3 right here, maybe it, instead of activating a transcription factor that turns something on, it could actually activate a repressor. Repressors bind DNA, and they turn them off. So it could have a change. It could actually turn off genes at this stage. It doesn't always have to turn them on. Over here, for example, K1 maybe, in a different cell, that's why I'm using the red, this could activate something like a kinase B. In this case, this could do something completely different. New cascade. So a new path could be forged in these cascade molecules. Up here you could have, the receptor could be a little different, and it could encourage a different kinase to bind, not just kinase 1, let's say. There's all kinds of ways that if you understand this pattern, you can add variables, you can say that it's activating multiple things, you can add a new signal that comes in, you could add another pathway that's like trying to deregulate this pathway. So if I give you a question to create that kind of scenario, illustrate the normal scenario for me, then show me ways that you can change it or add additional things. So the big important thing that we learned today is that it's a fight between repressors versus transcription factors, activators, and RNA polymerase, which is the big bad, that's the whole thing. This turns on a gene. So remember that these pathways don't always have to just be turning genes on in the end with their kinase cascades. They can turn stuff off. They can turn hundreds of things on. They can just form a new, different pathway in a different cell. And so remember, every time that you're working with one cell, think of a couple receptors that are going to get a couple ligands. Those might interact. But remember, in different cells even too, you may have a different receptor that binds the same ligand, has a totally different pathway. If you're still following along, just basically understand that I taught you this one way, but imagine it in different cells under different circumstances, as many possible ways as you can. That's what that practice question where I ask you to create a pathway, that's kind of what it addresses. And that will help you on the test get some points if you want to explore this more.